So welcome back to Neuroanatomy Part 2. Last time we went over some basic anatomy and physiology vernacular. We went over some basics of the nervous system, the peripheral and the central nervous system. We went over the autonomic nervous system and the different sub-branches of the peripheral nervous system. Today, we're going to be spending more time on the central nervous system and specifically nuclei that are within the central nervous system, like big brain regions that you probably heard of, like the amygdala and thalamus and things like that. We're not going to go super in depth into any of them just because I think that would be a little bit out of scope for this course to have you memorize all of the major subregions of the brain, just because there's a lot of them. And honestly, nobody is really an expert in all of the brain. You know what I mean? There's people who are experts in subregions of the brain, and then there are people who further specialize in the subregions of the subregions, right? So like the way that the brain is structured will become pretty clear in a moment here. But it kind of never ends, is what I'm saying. It's a rabbit hole of, if you're talking about the amygdala, for instance, then you're going to be talking about the central nucleus of the amygdala, the basolateral amygdala. Like, there's just like always new amygdalas. And I think I talked about that a little bit in one of the previous lectures, which is that the way that the brain is subdivided into certain structures is because of the functional nature of those structures, or because, well, not or, I mean, it's like and, because of the different physiology that's present at each structure. So it's not just that the amygdala or the cortex or the medial prefrontal cortex or the, I'm just listing just to give examples, or the prelimbic cortex. It's not just that they have unique behavioral characteristics, which they do, but they also have certain characteristics like pyramidal cells in a certain layer. They have granular cells. They have different types of cells present, which also defines the brain region. Or they have different molecular characteristics, like we'll go over certain areas later on where they only express, or they primarily express, dopamine cells or serotonin cells or whatever. And so because of that characteristic, it is divided into another subregion. But one thing that I think I probably mentioned in the last lecture, which is really challenging, is that people will have different naming cultures for the brain depending on their lineage of training and kind of when they were trained. So you don't hear too many people that were trained in my time period that are like referring to the forebrain. But a couple of, a decade or two ago, many people referred to the forebrain and the hindbrain. And I think that the reason why we don't talk necessarily about things like saying the forebrain is just because it doesn't really tell, like if you say that the forebrain controls X, that doesn't really tell you anything anymore, because there's so many subdivisions of each nucleus which you find in the forebrain. So it's really nebulous to say that the forebrain does X, because it's like, well, that's true, but are you talking about the cortex? Are you talking about the hippocampus? Are you talking about the amygdala? Which subdivision of each of these structures are you talking about? So all of this is to give you context for the way that we talk about the brain, speak about the brain in neuroscience. So one thing that's really frustrating other than that is just trying to figure out, the reason why the forebrain is called the forebrain, the midbrain is called the midbrain, and the hindbrain is called the hindbrain, is because of this structure right here that I'm showing you, which is the neural tube that your brain evolves from in utero. So initially, it starts off as just this little tube, and then as you're in the womb, the brain starts to take shape. And all of the brain structures arise from the forebrain, the midbrain, and the hindbrain. Obviously, with all of it, anatomy and physiology, there are other names for this, the Latin terms like the proencephalon, the mesencephalon, and the rhomencephalon. I'm not going to be using that nomenclature at all on the exam. I'll just say forebrain, midbrain, and hindbrain. But again, if you have any interest at all in progressing in, behavioral neuroscience, biological psychology, whatever you want to call it, then it is important to be familiar with these terms because people will use those terms. Periodically, you'll hear someone say proencephalon, although it is pretty rare, to be honest. So this is going to be a super fun lecture that's going to basically be me listing off of all the functions of these brain structures. So I know that it's not always great just to have people reading the information off the slides, but honestly, when it comes to this kind of thing, I'm going to extrapolate a little bit, but I'm going to basically just be telling you what you need to know. So the hindbrain is further subdivided into the medulla, the pons, and the cerebellum. And so we're going to start from the hindbrain, and then we're going to go all the way up through the midbrain and then to the forebrain. The medulla of the hindbrain, so again, remember the hindbrain is further subdivided into the medulla, the pons, and the cerebellum. We can see these things right here. And then obviously it turns into the spinal cord. But the medulla, or the medulla oblongata, is an enlarged section of the spinal cord. And the head and organs connect to the medulla and adjacent areas through the 12 cranial nerves that I'm pretty sure I've alluded to in the previous lecture, but we will talk about more in depth momentarily. The cranial nerves control a lot of the vital reflexes in your body, like breathing and heart rate and vomiting and salivating, coughing, sneezing, basically the things that your autonomic nervous system is controlling. And there's a large population of opioid receptors that are found here, which is the primary reason that people die when they overdose from opioid consumption is respiratory depression. And that's because there's such a big population of opioid receptors down here that can be targeted by opioids and cause respiratory depression, and literally the person will just stop breathing. So, opioids largely are central nervous system depressants. And so when you are taking opioids and you essentially the part of the spine here that is going to cause you to start breathing is shut off because it's so affected by the inhibitory effects of opioids. And when I say opioids, I'm referring to heroin, I'm referring to Vicodin, Oxycodone, obviously fentanyl, which people are probably more familiar with these days. But any kind of prescription pain reliever likely has some opioid component to it. The pons is also a part of the hindbrain, and it is Latin for bridge. And the reason why it's Latin for bridge is because this is where essentially the nerves are going to cross over. So it's important to know here that the nerves from each hemisphere cross to the opposite side of the spine, which means that the left hemisphere controls the right side of the body, while the right hemisphere controls the left side of the body. So yeah, that's an important aspect to know, which is that each hemisphere controls the contralateral aspect of your body. So your right side of your brain, you remember that your brain is bilaterally symmetrical, right? So each hemisphere has a mirror of the other hemisphere. So all the same nuclei are present in both hemispheres. And one kind of interesting part about the nervous system is that in the pons, since these nerves cross over and then travel to the contralateral hemisphere, that means that hemisphere is controlling the opposite side of the body. Contralateral meaning opposite side, ipsilateral meaning the same side. So if you have damage to the left side of your brain, you're going to have deficits on the right side of your body, basically. Whatever the organ system or somatic part of your body is being controlled by, whatever injured portion happens, it's going to be exhibited on the opposite side of where the brain injury is. We also have the cerebellum, which is it's referred to as the little brain, and it's not to be confused with the cerebrum. Okay, so we have the cerebellum, which is this little like small looking brain that's on the base of your actual, like the, this is part of your brain, so it's not your actual brain, but it's on the base of the cerebellum. And it controls like movement and balance. And it's one of the first things to start to develop and one of the last to complete development. And if you were to damage the cerebellum, you'd have a lot of issues with balance. And it also is interesting because it's important for learning. So it's not just a nucleus or an area of the brain that's important for somatic control, which it very much is. And it seems like that's like one of its primary regulators, or that rather the cerebellum is one of the primary regulators of somatic control. But it also has like, higher cognitive uh, uh, functions like in learning and memory and attention, and other social behaviors also, that which was very strange to me to learn. But one thing that's really interesting about the cerebellum, and this is the reason why I'm telling you this is because it kind of exhibits one of the really fascinating parts of the nervous system, which is that your nervous system is extremely plastic. And depending on when you receive damage to a particular area of the brain, your ability to recover and for your brain to compensate in different ways is really remarkable. Some people are actually born without a cerebellum, and these people show no to little deficits at all when it comes to somatic control. Like what we're talking over here, rhythm and the ability to judge time and balance. That's not something that these people deal with, even though they don't have the cerebellum. So what that means is that while the cerebellum canonically is important for, balance and things like that, other areas of the brain have the capacity to take over that job if it is the case that it happens early enough or you're just born without one and you have that kind of deficit. So this is a question that I will for sure ask on the exam, not just what the cerebellum is for, which I will, which is that it's for balance and it's not the cerebrum, etc. But that the nervous system in general, or that the brain is really plastic and is very good at taking over the job for a structure that is damaged if it happens early enough. When you're a kid or whatever, you are a lot more resilient than when you're an adult. And in fact, there's even a story from the 50s or 60s. I want to say probably more the 50s, because it was pretty terrible. What was happening was there was a psychiatrist. I mean, this was a treatment methodology in general. So it wasn't just this one. I said psychiatrist, but I meant neurosurgeon. It wasn't just this one guy that was doing this, but they were doing frontal lobotomies, which is where they basically take an ice pick looking tool and put it up your nose and then scramble the front part of your cortex, of your prefrontal cortex. And this was used as a treatment for a variety of psychiatric illnesses like schizophrenia, even severe cases of, what would now be called ADHD. And in particular, there's a story of a young guy who was 12 years old and had a frontal lobotomy and grew up relatively normal after that. So even though they scrambled his prefrontal cortex, at the age of 12, he grew up pretty much normal. And when I say normal, I just mean that like he didn't have all of the deficits that generally come with a frontal lobotomy. A frontal lobotomy normally kind of zombifies people. It takes away their affect and their personality and things like that. And it really, it controlled their symptoms in the sense that they weren't causing problems in these psychiatric institutions anymore. But it's really barbaric because it's really just causing severe brain damage to people. Anyway, the point of that whole rant, I think I may have gotten off topic here, is that the cerebellum is important for balance, and people born without the cerebellum, compensate for that and can still balance even though they don't have the main structure that's important for that. And furthermore, that's evidence that the nervous system in general is very plastic and can compensate for deficits if it happens soon enough or early enough in development. So these are the 12 cranial nerves, and I'm not going to have you memorize all of them, but you should just know that we have cranial nerves for a variety um, of functions, and some of them are redundant. So, we have olfactory, which is obviously going to be smell, the optic nerve, which is for vision, the oculomotor nerve, which controls eye movement and pupil constriction. And so that's one thing, like when you see people that have pupils, one pupil that's blown out and the other one that's not blown out. So you have a dilated pupil and a non-dilated pupil. That's like really a problem. That's typically indicative of drug use or some kind of brain damage, because pupil constriction and the movement of the pupils are tightly regulated by these cranial nerves. We also have the trochlear, which is control of eye movement, skin or the trigeminal nerve, which is important for skin sensations, control of jaw muscles for chewing and swallowing. And when you have damage to some of these nerves, like I'll get to that in a second. The cranial nerve 6, which is abducens. It's another cranial nerve that controls eye movement. And you have a lot of control over the eye movement, a lot of resolution and granularity over that, which is really interesting. For instance, if you were to shake your head back and forth really fast, you can still focus on the words on the screen. And that's because you have such control over where your eyes are going, that your nervous system predicts and then compensates for your head shaking, which obviously is really important when you're moving through the world to be able to compensate and continue to see where you're going. Versus if you were to shake your computer, or if you're reading a book and you shake the book, you can't really focus on the words, and that's because your nervous system is not compensating fast enough for what it cannot predict, right? So we have cranial nerve 7, which is important for facial. It's called the facial cranial nerve, which is important for taste for the anterior 2/3 of the tongue, control of facial expressions, crying, salivation, dilation of the head's blood vessels, etc. We have cranial nerve 8, which is important for hearing and equilibrium. We have the glossopharyngeal nerve, cranial 9, which is for taste, throat sensation, the posterior 1/3 of the tongue, taste, and control of swallowing, salivation, throat movement during speech. And then we have the vagus nerve, cranial nerve 10, which is the one that you will need to know about on the exam. It gives us sensations from the neck and thorax, control of the throat, esophagus, larynx, parasympathetic nerves to control stomach, intestines, and other organs. And so the vagus nerve innervates the gut, and the information that the vagus nerve gets from the gut is really important for control over feeding and hunger and satiety and things like that. So you will need to know essentially what I have written here about the vagus nerve. You will also need to know that the vagus nerve innervates the gut, and that the vagus nerve is important for a variety of feeding and hunger conditions. We have cranial nerve 11, which is control of the neck and shoulder movements, and then cranial nerve 12, hypoglossal, which is control of the muscles of the tongue. So you have 12 cranial nerves. You're not going to need to memorize all these, but it's just a few things that I focused on. Moving on to the midbrain. The midbrain is subdivided into two main structures, the tectum and the tegmentum. And for the tectum, it's broken up into the superior colloquius and the inferior colloquius. And the way that I remember this in undergrad was that I thought that, I don't know that I necessarily believe that this is true, but this is just what I associated with it, that vision is superior to hearing. That if you lose your vision, it's going to be a lot harder to get through than if you lose your hearing. I don't know if that's true or not, but for me at the time, I just thought that superior vision is, or vision is superior, either way. So the superior colloquius is for vision, the inferior colloquius is for hearing. And then we have the tegmentum, which we're going to get a little bit more in depth into right now, which has the reticular formation, the paraqueductal gray, substantia nigra, and the ventral tegmental area. So as I already said, the tectum, which is the subsection of the midbrain, is the roof of the midbrain, so it's the top part. And it has the superior colocus, which is important for vision, and the inferior colocus, which is important for hearing. And then we have the tegmentum, which, as I said, is broken up into these 4 different subregions. And the first one is the reticular formation, which is important for arousal, consciousness, sleep-wake cycles, coordination, cardiovascular control, and damage to this area of the brain can result in coma and complete loss of awareness. So the reticular formation is also what is going to wake you up when you are having some kind of an issue like choking or not being able to breathe. It will give you a stimulus so you can then wake up and come to. So that's something that you'll need to know, is that the reticular formation is really important for causing you to wake up in scenarios that might be life-threatening. And again, if you were to destroy it, then you would probably go into a coma. So we have the para-aqueductal gray, which is important for modulation of pain, your sympathetic response to pain, and learning defensive and aversive behaviors and pain perception. So actually, the para-aqueductal gray will facilitate or suppress pain. And physical pain is a big contributor to psychiatric disease states as well. So people that are in chronic pain are much more likely to develop anxiety or depression. So this is actually even an area of the brain that might have some therapeutic potential for behavioral disease states. Then we have the substantia nigra. For sure, you'll need to know about the substantia nigra. It is one of the 2 main dopaminergic nuclei in the brain. And the substantia nigra innervates the striatum and is important for movement and control of movement. And death of the substantia nigra causes Parkinson's disease. So, or Parkinson, I mean, this is kind of a philosophical issue, right? But is Parkinson's disease caused by the death of the nucleus, or is Parkinson's disease the death of the nucleus, right? But the important part here is that the dopaminergic release from the substantia nigra to certain movement pathways, like the striatum, is how you have fine motor control over your body. So when you have damage to release of this neurotransmitter, that's where the shaking aspect of Parkinson's comes up. So if you have too much dopaminergic release, which is something that happens with Huntington's disease, or if you don't have enough dopaminergic release, which is what's happening with Parkinson's, then you wind up both with a jitter or like an abnormal walking movement or gait, because the dopaminergic release is important for that control. So you need to know that the substantia nigra is important for control over movement, and death of the substantia nigra results in Parkinson's disease in general, but also Parkinsonian symptoms. So there is a group of individuals who were trying to produce heroin. Basically, it's a synthetic opioid, and they accidentally instead produced a nerve toxin, which, while it did target opioid receptors, it also basically killed dopamine cells. So the ventral tegmental area, which we'll go over momentarily, is the main area that releases dopamine in response to drugs of abuse. And so these underground chemists were making this drug that was basically targeting, dopaminergic nuclei, but it did target them, but it also killed them. So the people that were using this drug essentially were healthy, normal, young people with an intact substantia nigra, and then after doing this drug, it attacked the substantia nigra and basically killed it, and they immediately essentially had Parkinson's disease. So The substantia nigra, it's also called the substantia nigra because it's a black nucleus. So if you look at subsections of the substantia nigra, you can see a dark color to it, which is where the name comes from. So you need to know that that is the main dopaminergic nucleus that controls movement. And then we have the ventral tegmental area, which is the main nucleus for dopaminergic release in the reward pathway. So I say here that it's a key structure in the mesolimbic and mesocortical dopaminergic pathway. And whether you're saying mesolimbic or mesocortical, that depends on whether you're referring to the ventral tegmental innervation of the nucleus accumbens, which is in the limbic system. That's why it's called mesolimbic. Or the ventral tegmental innervation of the cortex, and that's why it's called the mesocortical dopamine pathway. So sometimes people will refer to this as the mesolimbocortical pathway, or the dopamine pathway, the dopamine reward pathway rather. And whenever drugs of abuse are used, then we get release of dopamine from the VTA to both the limbic system in the nucleus accumbens and the cortex. So that's for sure going to be on the exam, is that the ventral tegmental area is the other main dopaminergic nucleus in the brain. And the VTA, or the ventral tegmental area, is important for reward, while the substantia nigra is important for movement. Although I will say that in this part now, you can say this is not part of the exam, but just so you know, because it's important for how the brain works, is that it's very, like the dopamine that's produced here is not just for movement or just reward. It has a variety of mechanisms or functions rather. But the canonical view is that the substantia nigra is important for the control over movement, while the VTA is important for this reward mechanism. And just to illustrate the point, with the VTA, there was a experiment that happened to Chinese prisoners in China. So this was pretty messed up, but they basically were trying to cure drug addiction, and they had a mechanism of killing these dopamine neurons. And while these Chinese prisoners no longer wanted to do drugs, they basically didn't want to do anything anymore. They were completely anhedonic, and anhedonia means that they lacked any kind of reward or drive to do things that are typically rewarding. So if eating donuts makes you happy, when you're anhedonic, eating donuts does not make you happy. So it seems to have taken away their desire to do drugs, but it also took away their desire to do basically anything else. So not a good treatment for drug addiction. Okay, so moving on to the forebrain. It's the most prominent brain area in mammals and birds. Again, there's two symmetrical cerebral hemispheres, and it's organized to receive sensory information from the contralateral side of the body. So I already talked about this, but in the hindbrain, when we are talking about the pons, the nerves that are coming up through the spinal cord, they flip to the opposite side of the pons, right? So they're coming up the spinal cord, and then once they get to the pons, they flip. That's essentially what this is talking about here. That not only are you receiving or sending, information to the contralateral side of the body for your movement and things like that, but also you're receiving information, sensory information, from the opposite side of the body. And when we get into sensation and perception, we talk about vision and hearing, that will come up again. So we have the cerebral cortex here, which is this big portion of brain, which is honestly a lot of what people think of when they're thinking of the brain. They kind of think about the cerebral cortex. We're going to get into that a little bit more in depth when we talk about the different lobes. But the rest of the brain, essentially, all of the nucleus accumbens, the hypothalamus, the thalamus, the amygdala, all those things are found underneath the cortex. So we have the amygdala, which is important for evaluating emotional information like fear and anxiety. It's part of the limbic system. It's something that I think a lot of people are familiar with when they, the like one area of the brain that you probably have heard of is the amygdala or your prefrontal cortex. So the amygdala is a very important regulator for fear and anxiety, and that's what you'll need to know for the exam. But just in general, it's also has a role for feeding and reward and a variety of different things. The thalamus is a pair of structures. The thalamus and the hypothalamus essentially are a pair of structures in the center of the forebrain. Most sensory information goes first to the thalamus and is processed and sent to the cortex. So a lot of the information that's coming in telling you about the temperature or a variety of sensory stimuli, it goes first to the thalamus and then it goes to the cortex for further processing. Olfactory information is an exception to the rule where it goes directly to the cortex. Many nuclei of the thalamus receive their input from sensory systems and project to a particular sensory cortex. Yeah, so it's like, all of the sensory information comes to the thalamus initially. Like when you're seeing or hearing, not smelling, because we just said that olfaction is an exception to the rule. But the rest of them do initially go to the thalamus, and then it goes to the visual cortex or the auditory cortex, or the somatic sensory cortex. And a very interesting thing about the thalamus is that it's also important for working memory. So working memory is, we'll go over this in our learning and memory module, but working memory is essentially a form of short-term memory. So if I say, remember the following list of numbers, 783-732, you'll be able to remember a portion of those numbers for a short period of time. And as I'm talking right now, you're probably already forgetting what those numbers were. But if I said 783-382 or whatever, and I said, just remember that working memory that you're going to use, because maybe you need to use it for a key code or something, is going back and forth from the thalamus to the cortex. So thalamus is really important for a lot of basic, autonomic functioning, but also when it comes to cognitive control over things like working memory. The hypothalamus and the pituitary gland is a small area below the thalamus. So, like I said in the previous lecture when we were going over various anatomy and physiology terms, I don't know if I mentioned hypo and supra. I hopefully did, but hypo means below. So basically what we're talking about here is that this isn't necessarily the thalamus, it's just the nucleus that's directly below the thalamus. So it's the hypothalamus. It has widespread connections throughout the brain and coordinates incoming signals and controls homeostasis. So we're going to talk about the hypothalamus a lot more in our homeostasis lectures, but in general, you want to think about the hypothalamus as a regulator of the brain and essentially the autonomic output to control a variety of things like temperature and things like this. Damage in the hypothalamus leads to dysfunction in motivated behaviors like feeding, drinking, temperature regulation, sexual behavior, and fighting. And it modulates the pituitary gland, which releases hormones. So the pituitary gland is very important for a variety of things like puberty and things like this, and the hypothalamus controls the pituitary gland. So the main things that you'll need to know about the hypothalamus is that one, it's important for homeostasis, and two, that it controls the pituitary gland. We also have the basal ganglia, which is another grouping of nuclei. It's found lateral to the thalamus, and it's composed of the caudate nucleus, the globus pallidus medial, and the putamen, which is more lateral. It's important for movement, regulation, and integrates motivational and emotional behaviors for action coordination. And it's important for gradual learning of skills and habits. So it doesn't say it here, but it also controls or contains the striatum, which is part of the greater basal ganglia structures. So what you'll need to know here, though, is that the basal ganglia is important for movement regulation. Another one that most people probably have heard of is the hippocampus. It's a large structure between the thalamus and the cerebral cortex, and it's crucial for memory, especially declarative memory. Again, we'll get deeper into the different types of memory in our learning and memory module, but declarative memory is something that you can talk about. So the fact that you can say that Disneyland exists, the fact that you can say that you went to Disneyland when you were 7, those are both types of declarative memory. We'll explain the different types. Those are 2 different types of declarative memory, but we'll talk about that in learning memory. But you need to know for sure that the hippocampus is important for your memory systems. And I have here keeps track of where you are and where you go. This is something you also need to know, that the hippocampus is really important for your spatial memory. So, for instance, this was done a long time ago, so I don't know that this would be replicated with like Uber drivers today, but taxi drivers in London were studied a couple decades ago, and they were found to have enlarged hippocampi. And that was because they had learned London, which is a particularly difficult to learn city spatially, evidently. But so you'll need to know that not only is the hippocampus important for like declarative memory, personal memory, that kind of thing, but it's also very important for spatial memory. So for you to learn your environment, or for instance, maybe you're sitting in a coffee shop and you have parked in a parking lot that was not directly in front of the coffee shop, but maybe in the next parking lot over or something. Your ability to think about how to get back to your car is something that the hippocampus is heavily mediating. So, or maybe you have, maybe you're in your apartment, and where you park for your apartment, you have some parking structure, and you have to like walk down some stairs, and then you have to walk up some stairs, and then you have to turn left, whatever. That kind of a system is heavily modulated by the hippocampus. So those 2 things you'll need to know. And I don't have it here. I keep saying it because since I don't have it directly on the PowerPoint, I just wanted people to not think it was unfair, that you need to know that the hippocampus is important for spatial memory. And then we have the ventricles. So the nervous system begins as a tube surrounding a fluid canal. So as I kind of talked about earlier, this is the neural tube. That's how the brain starts off with. And within the neural tube is this fluid-filled canal, which become the ventricles. So the canal persists into adulthood as the main canal in the center of the spine, and as the ventricles, like I said. There are 4 fluid-filled cavities within the brain, which are these ventricles here that you can see. And each hemisphere has one of the 2 large ventricles toward their posterior. They connect to the 3rd ventricle. So we have these 2 lateral ventricles, which are the big ones. And then we have 1 third ventricle here. And then we have the 4th ventricle that goes down and eventually empties into the spine. The cerebral spinal fluid is produced in the lateral ventricles, and then it flows towards the back. So it is produced anteriorly, and then it flows posteriorly throughout the brain, and then it is recycled. Let's see here. 3rd ventricle connects to the 4th ventricle in the center of the medulla. Yeah. So let's see if I'm just not trying to not get ahead of myself to see what I talk about here about the CSF. So the fluid that I was just referring to is CSF, or cerebral spinal fluid. It's similar to blood plasma. It flows from the lateral to the 3rd and 4th ventricle, and then into the central canal of the spine. So like I said, it's produced here in the lateral ventricles, and then it flows in one direction, basically. It's also in the narrow space between the brain and the meninges, which we'll talk about like in a second. The membranes that surround the brain and spinal cord, that's what the meninges are, but we're going to talk about that momentarily. The cerebral spinal fluid cushions the brain against mechanical shock. So essentially, your brain is kind of floating in cerebral spinal fluid. So it's like you have your skull, and then you have, it's kind of wrapped around by these meninges, and essentially within that is the cerebral spinal fluid. So it is supposed to kind of help. Well, I don't know if it's supposed to. I mean, evolutionarily, the advantage is that when you have a head injury, or when you hit your head, instead of it just slapping against the skull, it's got some cushion with the surrounding fluid. If the cerebral spinal fluid is obstructed, it accumulates in the ventricles, and when this happens to infants, it's called hydrocephalus, and it can cause severe cognitive impairments. So basically what happens is, if the cerebral spinal fluid can't flow the way it's supposed to, it will enlarge the ventricles, and it causes this hydrocephalus, which is basically, it's the ventricles are expanding and not allowing the other areas of the brain to develop properly. So you have some severe cognitive impairments when that happens. And surrounding the brain are what's called the meninges. So this is a cross section of the skull, and we have obviously our skin or our scalp, and then we have a small area of tissue beneath that, and then bone, which is the skull, and then the meninges, which are the dura, the arachnoid membrane, and the pia. So what you need to know about the meninges is just that it exists, that there are basically this surrounding tissue that's rather thin. The dura matter is very thick. The pia matter is not as thick. And then we have the arachnoid membrane, which is in between the two and is filled with cerebral spinal fluid within the subarachnoid space. So, but basically the main thing that I just want you to know that you have meninges. And when somebody has something like meningitis, that's an infection of the meninges. And the meninges don't just surround the brain, but actually surround the spinal cord as well. So, and the meninges also are filled with the cerebral spinal fluid. And then we have the cerebral cortex, which is, again, the outermost layer of the brain and is super developed in humans compared to non-human animals, especially compared to non-primates. So primates have a developed cerebral cortex, but not to the same extent that humans do. And a lot of what we think our cognitive capacity is derived from is our overdeveloped cerebral cortex. So the cerebral cortex is composed of the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. And before we talk about those, I'm just going to briefly talk about the corpus callosum, which is a bundle of axons that connects the two hemispheres of the brain. So as I've said about 50 times now, I think, between the first neuroanatomy and this one, the brain is bilaterally symmetrical. So you have two hemispheres, and each hemisphere has all the same nuclei. They're kind of mirrors of each other. And the corpus callosum is the bundle of axons that communicates between the two hemispheres. And this is really important for just overall functioning. You can live and be mostly fine if you cut the corpus callosum. The brain will adapt to that. But it is normally an important connection. Information goes back and forth from each hemisphere through the corpus callosum. So you just need to know that the corpus callosum is how the two hemispheres communicate, roughly. Okay, and then, for the various lobes, we have the frontal lobe, which is important for voluntary movement, attention, short-term memory, planning, and speech. We have the parietal lobe, which is important for proprioception, language, and language processing. So proprioception is like your ability to know where you are in the world, so you can feel your hands out. If you close your eyes and you lift your hand up to the right corner or whatever, the ability for you to kind of feel and know where your hand is in that scenario is an example of proprioception. The temporal lobe is important for auditory information, interprets sensory input into meaning. So, the ability to hear a firework go off and then know that that was a firework, or to see a rock drop and to hear the rock hit the pavement and then know that information was a rock hitting pavement. That's all happening in the temporal lobe. Language comprehension is happening in the temporal lobe. And then the occipital lobe is where visual processing occurs. So your primary visual cortex is in the occipital lobe, and which is kind of weird because obviously your eyes are up towards the front, and then they send the nerves back to the occipital lobe to interpret that information. So overall, you should know these bullet points about the cerebral cortex. So I'm not going to. I might not say exactly voluntary movement, but I might say something on the exam like, the ability of you to grab a water bottle is, a function of which lobe, the frontal lobe, parietal lobe, temporal lobe, or occipital lobe, something like that, in which case you should interpret that as voluntary movement and say that that's important for the frontal lobe. Oh yeah, and also they're all named because of the parts of the skull that cover them. So this is so there's quite a lot of brain regions, and there's quite a lot of functions for each brain region, but I tried to distill it down to just the primary function of each brain region. So we'll have around 10 questions, I think, on the exam based on this lecture. So the big take homes from each of the sub brain regions you should know. I am definitely somebody that has a bias for reward systems and dopamine. So you should definitely, I don't know exactly yet which other brain regions are going to for sure be on the exam, but I will tell you that the substantia nigra controlling movement and the ventral tegmental area controlling dopaminergic release that's important for reward is going to be on the exam 100%. Definitely the fact that the brain starts as a neural tube and that's where the names come from will be on the exam for sure. So I know that this is a lot of content. There's a lot of different areas to memorize and to think about, but I think that I've distilled it down quite a bit from what it could be. So good luck, and we'll see you in the next lecture.